Hey guys, welcome back. Hope you all are doing well. Welcome back to week 2. This week, we're going to continue and finish up our investigation of enzymes and discussion of enzymes. And you've been reading about them, watching some videos about them. You're going to do an assignment, an experiment at home using some at-home materials. Excuse me. What you're going to need is a potato. I've cut a piece of potato right here. Any size will work. Any potato will work. Find a potato at home, cut off the skin, get a piece of potato. Second thing you're going to need is some hydrogen peroxide. You can get this anywhere. It's really cheap. You don't have any at home. Super simple to grab some from Kroger's or grab some from Walgreens or something like that. And then you're going to need a clear container. I've got a small glass here. Plastic will be fine. Okay, so enzymes are proteins that catalyze chemical reactions. And they're going to cause reactions to happen in cells. Enzymes are very specific to their reaction that they catalyze, and which means that only one enzyme will catalyze one type of reaction. Enzymes are really important for cell functioning. They cause cells to function and change rapidly, maintain balance, etc. All right, so you're going to investigate a chemical, an enzymatic reaction this week. So I'm going to take my peroxide and put some hydrogen peroxide in this cup here. Take my potato, I'm going to drop it in, and I'm going to look at what happens. So let's watch. What do you notice is going on? Okay, if you're looking closely, and you should be able to see that just fine, you're going to see a bunch of bubbles being released. And so you might think that that is the potato breaking down into hydrogen peroxide, but that's not what's going on. Okay? You can actually see a lot of bubbles coming off the sides as well. Okay, what's going on is there's enzymes in those potato cells, and this enzyme that we're looking at is called catalase. Catalase converts hydrogen peroxide to water and oxygen gas. And many cells, many different organisms have catalase because hydrogen peroxide is toxic to living cells. It'll kill them. But they encounter hydrogen peroxide often, and that chemical is made just simply because a lot of organisms use oxygen. And peroxide is water plus an extra oxygen, H2O2 instead of H2O. Okay, so what this enzyme will do then is break apart that hydrogen peroxide and release oxygen gas. So the enzyme is catalase, the substrate is hydrogen peroxide, and the results are that gas you see leaving, which is oxygen gas, and water. And that reaction will occur as long as there's a substrate. And again, that substrate is peroxide. And that enzyme will be able to break down hydrogen peroxide as long as there's peroxide available. And those enzymes aren't going to get used up. So you could take that potato out, put it in more peroxide, and that reaction will still occur. Okay, it's going pretty good now. You can see those, lots of those. But we get a good handle on how that reaction is occurring, how it's going, because of all those bubbles and how fast those bubbles are being produced. Now, what your task for this week is, find some way to modify that reaction. Okay, find something to investigate, some variable to change, and determine the effect of that variable on that reaction. So maybe what happens if temperature changes? What happens if pH changes? Do other vegetables, other living food, do other cells have catalase? So get creative, make it your own. Okay, so that's your task for the week, is to find some way to modify this and investigate the effect. Okay, and you're going to be able to determine the effect by looking how many bubbles are being produced. More bubbles, Less bubbles indicates speed up of a reaction or slowing down of the reaction. So once you've got your investigation mapped out, I just want you to submit a quick paragraph about what you did and how you did it. Make sure you include a hypothesis and an if-then statement. Explain why you chose the variable that you did. Okay, and do a little background on it. You should be able to find things that change chemical reactions with enzymes from your reading and from videos. You should be able to find those things pretty easily. Okay, other things in your short little write-up, I want you to have just a quick explanation of the results, and then apply that to enzyme reactions in general. So for example, if I changed this and in some way and the reaction slowed down, okay, I would then apply that to a broader scale. All enzyme reactions slow down because of that variable. Okay, so have a little fun with it. Make it your own. Get up, get moving, do something, some science at home, and then include a picture of you completing the investigation. Upload that picture for me, or a short video. I want to share that, and I want to see you doing it. So make sure that in your submission, you'll see this on Canvas. You got a short little paragraph write-up, and then include a picture or a short video of you doing your investigation. If you're having issues getting those materials, just let me know. We can work something out, but they're pretty easily accessible. If you guys have any questions, shoot me a message. I'll post those questions on the discussion board for everybody else to see. And get up and get moving this week, and let me know what you need. And we'll look forward to seeing you soon.